together with his colleagues Fateme and my colleague Emma Takorshit, and I will present it now. Just because I forgot to ask Sen-Yung in time, and so I hope I don't maim it too much. And but Sen-Yung promised me that he helps me with the questions, and so this is actually some nice work Sen-Yung had basically developed in our group, the method of calculating from steered molecular dynamic simulations potential of mean force or thermodynamic potentials. He applied it first together with Emma Takorshit and others to calculate the potential inside of aquaporin channel. That will be covered tomorrow in Emma Takorshit's lectures. And he then, one of the participants told me that she read this paper, she said something like 1,000 times or something. And we felt actually ourselves that that first paper was a breakthrough calculating with potentials, but we owe to the community a second simpler paper. And now Sen-Yung and his friends have actually now applied the method mainly to be pedagogical, explain it, but nevertheless it makes a paper that will soon come out in Journal of Chemical Physics to really very nicely, cleanly explain the method as a simple system. And actually, Sen-Yung also wrote a wonderful tutorial that you probably will find outside. Is it available now, the tutorial? Is it printed? Okay, so you will get it then in the labs, and that will be your second tutorial today. And so it will be not hopefully making it necessary to read it 1,000 times, but hopefully only 10 or maybe even only 2 times. So this is Sen-Yung's lecture. I just took his verbatim, his transparencies. So he applied this method to a very simple system that is actually, as I said, even for researchers, nice to read about, and so it will be in Journal of Chemical Physics, but it's also a wonderful example for the workshop. Now, the problem in somewhat more abstract language that we are facing when we do steered molecular dynamics is we transform the system from one state to another state. And here we denote it through this ellipsoid. One time it's like this, and one time it's always like this. But this, you could imagine that this reaction coordinate here is not the deformation of an ellipsoid, but is stretching a protein out, pulling an ion through a channel, and so on. And one would like to calculate the work in going from this point to that point. And we do it. We do everything fast in molecular modeling because we don't have the computer resources to do it slowly. And if you do things fast, you have to count on doing irreversible work, so that this famous law of thermodynamics, that the work you do in transforming a system from A to B is always larger equal the free energy difference in going from A to B. And since we do it fast, you would expect that this inequality is really always a real true inequality, that the work here is larger than the free energy difference. So then you say, how can you learn from this transformation, steered molecular dynamics, about the free energy difference when you always have some irreversible work, you don't know what it is? Now it turns out that even a field as old as thermodynamics, science is never standing still, can still develop. And there was a wonderful development to this field by Chris Jarzynski, actually a young physicist, now at Los Alamos National Lab. At the time he was in Seattle when he discovered this, and he found this equality. And this equality is actually the more general equation to this inequality. If you just take the property of the exponential to be have a concave shape everywhere, you can quickly derive that this inequality is a corollary of this equality. With other words, if you are transforming the work from here to there, many, many times as quickly as you want, and you do it and you sample enough processes, you can get the free energy difference. Now, whenever you hear the word sampling, you are right to get nervous, because you never know what that means. How much sampling? Is it, simple, you do 2 or 3 examples, or do you have to do billion times zillion examples, so you cannot really do it? So that didn't come with this equation. So one didn't know how easy it is actually to apply this equation. Now, before I go on, I would like to quickly tell you, with Sun Jung's words, where this comes from. So if you have a Hamiltonian system, then you do external work. You can calculate this. We have a Hamiltonian system that's time dependent, the charm force field, time independent, and then we apply a force that is time dependent by pulling something. And so now we have the time dependent Hamiltonian. We can calculate the work. And now this Jarzynski equality holds as long as this process is Markovian. That means that we have an equation of motion that is of this type. And we have here an operator that is only acting at one moment in time. And I don't know how much of you know the various formulation of classical mechanics, but you can convince yourself that classical mechanics obeys this kind of equation. You just have to write here the Poisson bracket as an operator, and then you can show that this is just a classical equation of motion can be written like this. Now you also must satisfy the detailed balance condition. That means you must have a situation that you have an ergodic system where the Boltzmann distribution is actually an equilibrium distribution for the time being a fixed parameter. And so again, you can show that this is true for a classical system. You learned that in statistical mechanics. You can also show that this is actually true under the various simulation conditions that we apply when we add frictional forces and other forces to keep pressure constant, temperature constant. These 2 conditions still apply, so that we can assume that the Jarzynski identity holds, and we can use it for the analysis of the data that one gets out of steered molecular dynamics simulations. Now, however, comes a problem, and that is actually the only real intellectual thing you need to understand, except that you should register that the famous inequality is actually a corollary for many types of dynamical systems of an equality. But here, we have to calculate so if we just apply logarithm, we can write the free energy that we want to calculate as this expression. And now we need to calculate this quantity, sample this quantity well. And now let's say the W, the work that we do as we pull several times the system from A to B, let's say the W is distributed according to this black distribution. Then the distribution of the exponential values is this red curve. Because as you can see that when W becomes negative, it really blows up this value. And so to the negative side of this distribution, you're getting here this distribution of values for the exponential. And now this is what you sample. You do work. One time you do this work, one time you do that work, and this work, and this work, and this work. Very rarely do you do this work. Almost never do you do this work. Even more rarely you do this work. As a result, you're not going to sample well the exponential. So you're getting a huge sampling error there. That's a problem. I mean, huge, maybe I shouldn't say huge, so without qualifications, but you realize that you're having a sampling problem, because you have to sample well rare occurrences of the work. Now, you can now be practical minded and say, so I have a huge sampling error for the exponential. Maybe I play with accepting, instead of a sampling error, an error connected with a mathematical approximation by doing an expansion of this operator, so-called cumulative expansion. So you do the cumulative expansion just like a Taylor expansion in terms of power, it's a little bit more intelligent than a simple power expansion. And so you do this cumulative expansion, and now you realize that if this is the distribution of W's, the distribution of these moments here, the second moment, first moment, third moment, is given here by this green curve. This is the second moment, the blue is the first moment, and this violet is the third moment. You see they are much more centered where the W values are more probable. And so the sampling errors of these ones are smaller than of this one. So now you have a balance of a statistical or sampling error and the truncation error. And now you can hope that the truncation error is smaller than this error, or that the balance is in favor for using this expansion. And that was the idea that Sang-Jung and friends tried out. And so now we apply this idea, and you will do that too, to this stretching the helix. So you see there's the alpha helix. Deca-alanine is known to be also on a vacuum condition, a nice alpha helix. You stretch it out. And the nice thing about the system is so small that nobody knows what the potential of mean force is, but in fact, you can calculate it precisely. And that you see, so here what you do is you're pulling, sometimes you're stretching it out. This is the end distance, and this is time. You stretch it out, slow, medium, fast. This is the force you see. And if you calculate from this trace going forth and back, the potential, applying this identity, you find you're getting here a reversible, we're going forward and backward, you're getting, when you do it slowly, the same energy. And so that tells you that this is the correct potential. So you can do it so slowly, forth and back, that you can calculate the energy at each moment of time. You don't get any hysteresis, and so you say, okay, this must be then the correct energy. And so now you have for this system a gold standard. You know what you have to aim for, and now you can play around, pulling it faster and see what error you make actually, and then you can know, what truncation error, how far you should expand, how many samples should you take, and that is what you play through, and that is what Sang-Jung and friends figured out. And though I just want to show you what their results are. So here you see the potential that is connected with the stretching out, and they found actually that this increase is mainly connected with breaking hydrogen bonds, so the hydrogen bonds come in again. And now you see here different scenarios. Here, this is the intermediate pulling velocity, and you see that if you do first order approximation, you have too much of a rounding error. The statistical error is small, but the rounding error, truncation error is big. Second order, smaller truncation error, little bit bigger statistical error, but you see you're getting a very nice reproduction of the exact potential. If you go to third order, now you're making a too large of a statistical error, and so that is not good anymore. And if you take the exponential average, then you see you also don't do as well as if you do the second order cumulant, because you have too much of a statistical error in the exponential average. This is using the identity directly. And so from this you see that second order cumulative expansion is giving you reasonable values. Now if you do a faster pulling, then you see you quickly deviate. Second order you still do pretty well, but of course, depends on how ambitious you are. Here you're getting huge errors. Exponential gives you a big one, so you see again, even at fast ones, the second order equivalent is pretty good. Now there is a variant that has been mentioned so many times, because this is what people have been doing for many years, and that is umbrella sampling method. Basically what you do is, as I described, you pull to a certain place, and then you basically hold the system fixed, but with a little wiggle room, so that the end can wiggle. You have basically a little spring there, and you see that the distribution of the ends is then given by this. Then you pull further, and then a new equilibrium position is like this, and here you see all these distributions, and here you do that by sampling less often. So here you sample 100,000 times. It's the same computational effort as pulling with the intermediate velocity, and this one is the same computation error as pulling with a fast velocity with steered molecular dynamics. And here you can construct now with this umbrella sampling method a potential. I tell you quickly how this works. And here you can do it again, and you see here you're getting, this is intermediate velocity, and this is about as good as the other method. And so you cannot really say umbrella sampling is better or worse. And I just would like to tell you that the only argument you could possibly give against the umbrella sampling method is that what you actually do is you have a trajectory, and let's say this is a time window where you have pulled up to a certain place, and now you sample here how the system wiggles. You're getting a distribution out of it, and now you use this distribution here, so like the trajectory where you're getting your distribution. You have a distribution here, you have a distribution here, you have a distribution here, and now you have this independent distribution that you sample at 10 different distances, for example, and each of them would correspond sort of to a Boltzmann distribution, but you would like to get a Boltzmann distribution that governs all of them. So you're getting one Boltzmann here, one Boltzmann here, one Boltzmann here, but you would like to now get the Boltzmann distribution. This one is a Boltzmann distribution of the real system plus the superimposed potential. You would like to discount the potential that you use to constrict the system, and you would find then the underlying potential that is the best underlying potential interpreting the remaining features of this distribution. And the way you do this is basically by measuring this individual distribution P0I, and then you have here something that you can easily calculate. I don't want to go into detail, except this ratio here. For this ratio, you need a factor here, so everything can be, this is your sample here, this is the number of data points you take, but here you get a factor that you only get if you know already the answer. So you need a factor in order to get P0, which is the overall potential of mean force. You can get P0 only if you know P0 already. Now we all know how we do this, we do it iteratively. And this, that you call the weighted histogram analysis method. It's a certain method that makes certain statistical assumptions about the statistics of this thing, and then it can find the best overall Boltzmann distribution that is in harmony with this individual distribution that you measured. And but you see, you have to do an iterative calculation, and now you can judge yourself, you know, is this so awful that you want to do the other one, or is it equally good? And I think basically there is not much of a difference. I think the main thing was that steered molecular dynamics, when it came about, was really the bold method that people realized you can operate off equilibrium, you can do non-equilibrium processes, you don't have to worry about it as much as you were told. This method actually assumes underlying that you are always in equilibrium, and so people thought, oh, we have to be in equilibrium in order to make the system work, and they didn't dare to do anything bold like manipulating a protein around. But now, as we see, this compares well with other methods. Other method is a non-equilibrium method. You can be bold, you can play around, and so I think it's basically more an attitude than really an mathematical difference that we are talking about here. Okay, so attitude, playing around with the system. One of the key things in doing steered molecular dynamics is that it's so natural that you would like to do it in real time. And that is actually possible. You will actually do that yourself this afternoon, because we have taught you about the graphics program, we have taught you about the molecular dynamics program. Why not link the two together? Why not do a simulation and watch the simulation as it is happening on your VMD screen? And that you can do, and the advantage of it is not that you now see the protein wiggle, because you don't learn so much from a protein wiggling. You can now manipulate the protein. Now with the mouse you have, like it's a little bit limited because it's a two-dimensional device. You can pretend it to be three-dimensional a little bit. But the best way to do it actually, if you would visit us upstairs where we have what is called a haptic device, it can give six-dimensional input. You can go to every point, every atom in three-dimensional space and pull it in any three directions you want. And you can feel even the force that the atom resists to you when you try to pull it. And so you can really then pull one atom, several atoms at a time. You can define that quickly in the computer and manipulate this with something there where you have a very natural mechanical feel. But you can do it with a mouse. You will do it with a mouse. You will see that very natural to connect VMD with the molecular dynamics program and then have the two talk to each other. You will see more of it also tomorrow with in Emat's lecture about channels, but we just would like to give you, because it's sort of fitting with on the SMD day, we want to show you here a little bit how it works, and we just take the little silly example, the buckyballs, and now I give you a microphone, Jordi, and then you can tell us what you see, and then we have our question-answer period.
All right, thanks. I didn't know I was going to talk. Okay, so here what I have is a system that's running on one of our clusters. So it's running on 10 processors, and it's just a simulation, but I've connected it to VMD so I can see what I'm doing. And these are just unconnected graphite sheets and some buckyballs. This is just a simple system. It's kind of whimsical and it's fun. So right now with the mouse, I can apply forces, so it actually feels what I'm applying to it, and I'm sending those forces to NAMD on the processor, so I can actually interact with the system. And the system is fun because okay, so now it's running away. Okay, so I'll take another one and I'll collide them together.
Actually, it happens all the time.
Just having a little bit of fun. So actually, I'll do some little things, but I'm done here.
No, do one more. Bump
yeah.
One into the
but you
plane.
can talk, but I'll do stuff. Okay.
Okay,
and you will get to try this maybe today and a little bit more tomorrow. And this is actually fun. You can do this on your local machine.
When you do this stretching, then you can look at this one. Look at it, how it bumps in there. Okay.
You can do this with protein too. You can do this with scientifically relevant systems too.
So now you see how SMD really works, and I'm sure you will trust it. Okay.
That one's gone. All right.
Okay. Okay. Thank you very much, Jordi. Okay. So we have now the general question period already. Let's do that. And any special questions about the lecture, also fine. Everything is clear. Oh, there.
How do you take into account the number of atoms in your system and the speed of the processors and the number of processors and figure out, how do you calculate how long your simulations are going to take?
Yeah. So it depends on three issues, or let's say on four. So the first one is, what is the relaxation time when you set up your system? What is the relaxation time that the system equilibrates? You might set up a system in a way that you are worried that it may take a long time to equilibrate. For example, in a membrane, the lipids may have to move around, or ions may have to move around, or you have modified the protein because you want to try out another structure. And so you have to know you have to make a best guess of that relaxation time. Let's say it's 10 nanoseconds, then you have to equilibrate already, let's say 20 nanoseconds. If you have a good feeling that it's only 100 picoseconds, you may just equilibrate 1 nanosecond. So the relaxation time that governs the equilibration. Then you have the process time. You want to study a certain process. Now, some processes, we of course prefer to do processes that are fast. Let's say you have a process that is known experimentally to be a few nanoseconds, and it happens in a few nanoseconds. We will be happy, we just do a few nanoseconds. If it's one that takes actually milliseconds, then either we give up or we say, let's try steered molecular dynamics to accelerate it, but we can do that only when we have an idea about the reaction path. So you have to develop an hypothesis there. You can modify the hypothesis as you practice it, but you can then possibly accelerate it. And then you have to accelerate it time that is available to you in terms of the computer power you have. Now that is a third factor, computer power. You know, we have now Lemieux, that is this big 3000 processor machine at Pittsburgh, but we can use that power only for, first we need to get computer time. You get twice a year opportunity to apply for it, and you need to get that computer time, but you can use that many processors only for large systems. For a system of 100,000 atoms or larger, you can use 1,000 processors, but if you just have 10,000, then you cannot use so many processors. The experience there is that you're better off using only 16 to 32 processors. And now, if you have those available in your own lab, you might have a cluster of 16 processors. Then I think you do a 10,000 atom simulation of 16 processors very effectively. You're losing almost nothing in terms of, you know, the parallelization. And then you go on for how long your colleagues in the lab let you use that thing, and until they kick you off. And yeah, so it's so in short, relaxation time of the equilibration, process time, possibly process time for an accelerated, for a steered system. You need have access to big computer, but depending on your size, you can actually only use effectively a certain number of processors. And so if I would scale it, I would say with 10,000 you can do 16 well, with 100,000 you can do 500 well, maybe 1,000. I don't know where you are. But if you are small, like you have something like 5,000 atoms, then you cannot gain much from parallelization. Then you just have to, you can use 4 processors, maybe not more. Maybe somebody's Jim here or somebody can quickly say, let's say we have 5,000 atoms and we want to not get less of an efficiency than 80%. My gurus, how many processors can we take? 4, okay, so 4. Okay. Okay, let's have the lady a second question, but I will not forget you. Okay.
Will you announce when the overheads are available online?
Yeah, the overheads will be made available pretty soon after the lectures. They can only be delayed when Sen-Yung Park, who is really doing that very, very well, when he has the help in the hands-on, then it could be delayed to this evening. But I think between now and this evening, my lectures will be online, and I'm pretty sure that most of the other lectures will also be online.
And what does MAMBI stand for?
Okay, guess. Only the people who know cannot. Okay, so it's a real stupid name. We're really deeply ashamed. Okay, so quiet. Not another molecular dynamics program. People who know Unix and computing know where this comes from. Okay. So there was a question, I didn't forget you, but this, and then you, and then from there. Okay. You have to speak up for me, I'm an old man, and also for the rest.
I have a question about this Jarzynski column.
No, the second. The second one. We thought that the second order term was actually better than the third order term. The third order term has a larger sampling error and gives us worse results.
Well,
Little bit like an asymptotic expansion in mathematics, you know, like when you WKB expansion, you also better stop earlier than too late.
But under the assumptions of Gaussian distribution, underneath Gaussian distribution for the dominant is okay. But I guess I'm wondering, how do we know the underneath distribution is?
Yes, actually, yeah. Actually, that's a good point. On the other side, it's not a critical point, though it's a good question you ask, and we should figure that out. But of course, we can measure the distribution of W. You know, we do W, we get the distribution, and we know what it is. So it might not be Gaussian, and of course, we also have to sample well. But I think we should, for the future, you know, pay attention. Always look what is a W distribution until we see certain patterns, and then we could say, they are maybe always Gaussian because central limit theorem, and we don't have to worry anymore. But maybe they have some strange tails, and so we typically use between 10 and 30, but here were sort of like little tricks made. You saw there's some block averaging and so on. But I think, let's say 20 is actually a pretty good number of what we did so far. But we don't have yet a large experience. So you can read the papers, they say it. And I think in the paper for the channel we did 14, but in a small interval, and then again, no, we did only 4. Only 4 in an interval, 4, but then only over a very small section. So this is a method that is being now applied for the first time, and we have actually only the second paper out, or soon out, that uses the method, and so I think time will tell.
As long as the inherent distribution is Gaussian, if it's not Gaussian.
No, that's a good point. That's a good point, and you know, like, of course, you only know the real distribution if you do really good sampling, but you know, like, some kind of sampling will probably give us over the next years a feeling for it. So maybe anybody who wants to use it later, maybe you also contribute to sample it, and maybe let the rest of the community know what you find. Actually, it was also applied by Bustamante from Berkeley in an experiment, because you know you can also do an experiment, and he did it, and of course he can sample more experimentally than we can do, and he sampled 100 trajectories to calculate the stretching of RNA, and so that's maybe another data point. Okay.
I have one question about this point. You want to use this situation for experimental data. Now we get results from numerical simulation. We can have as many samples as we need, for example, 1,000, 10,000 meters. How many samples can we get from atomic force microscopy? Is it because this is a mechanical property of the system, internal property of the system? Can you get, for example, 100 samples, 200 samples, or you can get, for example, 10,000 samples?
Yeah, it's that's a little bit that's really an experimental problem. So, like for the immunoglobulin domains, the way it works is that you actually do not connect the tip and the substrate covalently. You put it down, pull up, you pull up, and now you have a computer that looks at the result, and there's one type of result that looks like what I showed. That is a clear indication that you have a single connection. Either you have no connection, or you have sometimes a mud 3 or 4 things, but once in a while you get a nice connection. And so you do this, and you measure, and it works overnight. But you get very, very few good measurements. And so they can also only sample, value sampling distribution, I think probably also only 100, depends on how long you run the experiment. And of course you always need to refresh your solution and so on. So it's not just a matter of the computer running, but also of your sample staying alive. So also 100. So, but if you have, for example, a covalently connected tip, and you can do it, the only one who have succeeded that so far were some real patient, heroic Japanese who really, because it's very difficult to connect it. So you can go down and down 100,000, 10,000 times. But to covalently connect, you have to do chemistry and then you try. If you have just one connection, fine. If you have two connections, throw the experiment away, do it again. And so every time you do this, it probably takes a few hours. Maybe you can automate that too, but you know, you don't have that many atomic force microscopes. So that's very difficult. But once you have it connected, then you can of course pull, and then you probably can easily do thousand measurements.
I'm thinking during one trajectory, how many samples
Mhm.
take? For example, your stretch was floating for 100 angstroms. Are you taking sample every angstrom, or you can take sample less? I don't know how
Yeah,
many samples.
yeah, Bustamante, they really measured at a distance the force they applied, and then they calculated the work, and then they had actually the potential for every point for full stretch until full stretching.
How many points?
They took 100. Yeah, they took a smooth curve, of course. You know, they took many points, but then they pretty sure they must have chosen some function, like a spline fit or something that smoothed over the numbers they got there, and then so that I don't know how many points they could really do and what the scatter was, because I don't know if they published that. Mm-hmm. Okay, so you had a question, yeah?
Yeah, besides AFM experiment, can you simulate 100% nature process of DNA dissociation from double helix to two single strands with the help of enzyme?
So if I can do
Simulation of DNA helix stretched into two single strands by enzyme.
Okay, if I can do that, I not. My daughter probably also not, but maybe her children can do it. so I don't think we can do that at the moment. I mean, the whole process is very difficult, you know, like if you help it all the way you can, but there is there's a guy, Kevin Sobamatsu from Los Alamos. He used NEMD recently to simulate ribosome, 2 million atoms. And okay, wait, that was you were first, and then.
I just want to know how you apply precisely the force when you do those streaming experiments. Because, well, is it the center of mass that you decide and then applying the force in the center of mass? Are you distributing the force over the whole extremity of your...
No, we pull on one atom, and it is the connection between the atomic force microscope and the substrate to the protein is not so clear, and so that's actually what is being criticized about the experiment. Is non-specific. You just know that somehow it's connected to the substrate and to the tip. You know you have only one because you're getting one clean rupture and then it's gone. Whereas if you have several, then you're getting stronger force, and suddenly they drop, and you can get another cascade drops, and then you go down, and so you know have one. But how they exactly connect is not known. So we take just the termini, and we take the C alpha atoms, for example, or some other atom, and pull on both. But we also do often simulations where we pull on groups of atoms. And then we group, for example, we pull, for example, according to the center of mass. That's sort of good, because then the heavier atoms are pulled more than the lighter ones automatically, and so that's then a good choice of pulling. So if you have, let's say, 20 atoms, you won't pull them all, then you just pull the center of mass of them, so that the hydrogen is pulled little less than the other ones. Yeah?
Your group wrote such fancy programs for molecular modeling, for molecular visualization and molecular dynamics.
You said expensive?
No, not expensive. I said fancy. Nice program. Which would be used for molecular modeling? Because this, I've seen you didn't develop something for molecular modeling, so maybe you can advise us which one to use for molecular modeling. You don't need only download the files from as PDB files from the database, but you have to model.
What do you mean by, oh, you have to build structures or something? Ask my wife. That's her department. I stay out of it. And so she's giving you the lecture on Monday and Tuesday. I will warn her, but she is an expert in protein structure prediction and also in modeling proteins. And so she can give you a much better answer. Okay, with that cop out, there are some announcements.